Now start whenever.
Okay. Hello, my name is Brian, and I'm working in the Honda Medical Campus. And so the question that we are primarily investigating in this project is how cells are activated in the brain when mice are placed into a virtual environment. And so the cells that we're looking at primarily are in the hippocampus, which is this structure here, and it's shaped differently in most mammals, but this is a mouse hippocampus. And so this is an area of the brain that is associated with learning and memory. And so we want to see whether or not these cells that are activated, we want to see whether these cells are activated by this exploration in this virtual environment. And then we can perhaps form projects based on exploring how these cells affect learning and memory. And so the reason why we are interested in looking in exploring how virtual reality environments affect mouse learning is because using a VR rig is necessary in order for us to perform certain experiments. For example, optogenetic experiments. So in this case, sorry, let me go back to the first slide to
That's fine.
explain, yeah. The thing with all these experiments which require VR is that they need the mouse to be head fixed so that we can look into the brain or we can align a microscope right over the brain. And so we have the head fixed by these two plates while the mouse explores the virtual environment. And in the meantime, we can do things like look inside the brain, image cells, look at cell activity. And so the sorts of experiments that we can do with this rig are, for example, optogenetics, where we can go into the brain, cut it open, and then either excite or shut down these cells using light. And so some of these cells have these things called light-gated ion channels, which are activated by light. And so we can cause these cells to be more active or less active by changing how much light gets to that area. And another type of experiment we can do is calcium sensor imaging. And so in this case, we'll inject a fluorescent sensor into the area that we're looking at, and then when these cells fire or are activated, we can look through the scope and see like a bright red, like these cells become bright red because they fluoresce, and then we can monitor activity using that. And so what we're primarily looking at is how VR compares to, whether or not VR is like real world environments, in that does it stimulate the brain in the same way. And so we already know that when mice are exploring real world environments, they produce a protein called FOS, and this is a marker of cell activation. And so we know that if we can see FOS in these mice that are excited by the virtual environment, then we'll have the same type of activation as activated in the mice that explore normal environments. So these pictures turn out a lot darker than I thought they'd be.
Well, it's pretty light, so it's.
Oh, okay.
The one on the left's fine. I don't know about the one on the right.
Okay. Okay, I guess I'll have to fix this one. But here we have, we stain for FOS, and basically I'll explain the staining procedure later. But what we do is mark certain cells that we want to look for. And so these cells will appear darker when we look into the microscope, and we can count them or see how many active cells there were. And so this was supposed to just be a control slide, and so there was not supposed to be anything there. But it was just nice to compare the two. Unfortunately, it's pretty much all black. And so this is this type of staining procedure that we use to mark these activated cells. And so when we're staining for phos, we're looking for something that will make the cell, or make cells stand out that have phos in them. And so first we will attach a primary antibody to the to phos, and then we'll add a secondary antibody which will bind to the primary antibody. And so it's kind of like a chain sort of, except for with each step the signal gets amplified. And so maybe 2 secondary antibodies will attach to the primary antibody, which is attached to phos, and that continues building. And then we have another step, which is the actin-biotin enzyme complex. And so this continues to build, and so now we have phos, primary, secondary antibody, and then this ABC complex. And the reason for this is that when we are able to stain for the when we are able to attach a marker, the signal will be amplified because more markers will attach on the end of that cell. Like, for example, if it were just a primary antibody, maybe if we were just making the primary antibody fluorescent or something, we would see maybe like a small signal. But with all these segments, or sort of fluorescent proteins like attached on top of more proteins, the signal is amplified and we'll get a bigger signal. And so in this experiment, we decided to perform our experiment by first dividing the mice that we were going to experiment with into 3 groups. One that was placed in one virtual environment, one that was placed in one virtual environment, and then later switched to a novel virtual environment to see whether the level of excitation would be increased because they're now exploring more. And then lastly, a control group which does not explore. And so we would look at the levels of phos between all these brains, and from there we could learn some things. And so after we divide these mice, we give them head plate surgeries. And so what this is we will cut open the scalp on the mouse and attach a head plate with this glue so that the mouse can be placed on the VR rig. As you saw earlier, the mouse is fixed by its head plate so that it will be staring ahead straight at the VR screen, and so that we can, so the head is still so that we can look into the brain. And so after that, we will water schedule these mice, which is we will lower their body weight to a certain level by giving them less and less water so that they'll be motivated to do the VR training because they'll receive water as a reward. And we'll train them in the VR, which I will show in the next slide. And then after that, we will perfuse the mouse brain tissue, which is done by here, where we will puncture the heart and inject paraformaldehyde, or fixiv, which will cause the proteins in the body to cross-link. And so it basically preserves the body, it preserves the cells, or preserves the proteins in the body within the state as if the mouse was basically still alive. And then we can then stain for the phos that we're looking for and then take pictures. And so this is the sort of virtual reality environment that we are looking at, or that we use. And so it's a fairly linear path. And so they might walk from this end and until this end. And there are water rewards at each end. So every time the mouse runs to the end, they get some water. And then once they run back to the other end, they get some more water.
Are they on a wheel? Or?
Yeah, they're on a ball.
On a what?
On a ball.
Okay. Is that like a wheel? I just know the wheels.
So it's like the ball is suspended on a column of air, and then as they run along it, the ball.
Okay, so they can run on.
Yeah.
So it's a fancy wheel.
Basically.
It's not a wheel at all, but you know.
No, yeah.
Keep going.
And so, and they're different. We can change the environment with different textures, different colors, etc., and then see how the mouse responds to this. And so, although we haven't gotten the end data yet, we hope to see whether or not these cells are actually activated by this exploration by comparing the number of floss stained cells in each group. And so what we would see is that if there are more stained cells, these areas would be more, these brains would be more activated. And so what we are thinking that we'll see is that when brains is that the mice that have been exploring the virtual environments will have more floss and be more excited than the control groups, or the control group. And so the project is still in progress, as we still have to continue training the mice, and that's taking a while as some of the mice don't want to learn. But after we, if we are to see whether or not cells are activated by VR, we can look at using the types of methods that I mentioned before, such as optogenetics and calcium sensor imaging. We can investigate how cells are activated, or which cells contribute to learning, when we can turn off cells or activate more cells as the mice are exploring this virtual environment, and see how their performance does. Or, for example, we could look at how which cells are connected to other cells by inactivating them and seeing how other cells' activity is affected in response. And thank you for listening.
Good. So I think at the end your point, and correct me if I'm wrong, so once you understand which neurons are activated, or well, essentially are activated in response to different VR modalities, and I guess I don't know anything about VR, different VR modalities, you might be able to inactivate or activate specific neurons to see if those neurons modulate behavior X. Is that right?
Yeah.
Right, okay. Okay, good. Go to the first slide.
Okay.
I forgot to time you. You're a little bit long, but that's okay. I think you're a little bit long.
So there are a couple places where I put in parentheses I'll replace the picture. And I'm still kind of talking with my PI about putting in videos or other pictures.
Yeah, that was good.
Yeah.
That's okay. Yeah, so what you did, you went a little bit fast here. So how does virtual environment training affect? Just say the title. That's your title. That's a fine title. But what you did is you started to say the title, you clicked to the next screen, then you came back. And so I would just say, you know, our question is, how does virtual environment training affect brain activation in mice? Here's our general setup. I think it's fine to say what your general setup is, because then you have to come back to it. You know, you can show your whole rig. That one's not bad. I don't know. Okay, so that's what you're going to do. What's the next slide? Okay. Why do you want to do this?
Huh?
She's taking pictures of stuff I wrote.
Oh, okay.
Okay. Why do VR with this at all? So I agree. I mean, you have activity, and you said it later in the talk. You have your hippocampus. You do certain learning and memory, and it gets activated, and that's what's active. Okay. But why do you want to do VR in this?
That's so that we can look at how cells are. This is so that we can do those experiments that require the head to be fixed, or fit in a fixed place. And so, like, if we put them in a real environment, they're gonna have to wander around, and then we can't keep their head still, and as a result, we can't image.
Okay. Good. And in addition, it seems to me, so it seems like the first step here, and you said it later, but you didn't say it here, and I think you should say it here, is, I think the first step is you want to see, because it's already known that the hippocampus is activated, or cells in the hippocampus, I think, the way you said it, are activated in response to learning and memory, right?
Yeah.
So it seems to me you want to see if learning in virtual reality settings, do they faithfully mimic what we already know?
Mm-hmm.
Is that true?
Yes.
So that if they do faithfully mimic what we already know, you might be able to impose greater experimental control, i.e. we're going to turn this cell, let's say everything is the same, or mostly the same in VR, in terms of how the spatial, the kind of spatial learning and memory works. Then it seems to me you can then integrate optogenetics by turning these cells on and off and kind of try to get a better understanding of the cellular basis of learning.
Mm-hmm.
Right? Because, well, this one is activated when they turn right, and this cell is activated when they turn left. Let's optogenetically inhibit this cell when they're turning left, or let's optogenetically activate the right cell when they're turning left and see what happens. Mm-hmm. Is that the big picture? I'm trying to kind of hit the big picture.
Okay. Yeah. So what we want to do is that once we have these heads fixed, we can do these experiments where we, once we've confirmed that, or not confirmed, but once we've explored how VR affects these cells, whether or not they're activated or not, etc., then we can go in and, because these cells being activated indicates that learning is happening, and that it affects learning in some way. And so we would like to explore more how these cells affect this learning process, whether by inhibiting them or examining their connection.
Okay. But again, I think it seems, and you can check with Ed about this, it seems to me that the initial step is to see if the VR, and I think this is what you're doing, if the virtual reality setting, or how mice learn in the virtual reality setting, is a faithful surrogate or mimics, parallels how they appear to learn in a normal environment, right? Because mice don't live in a virtual reality environment, obviously. So it seems to me that this is setting a foundation to see whether or not mice learn in a similar way in virtual reality, and if so, it then allows you to have far greater experimental control over learning and memory experiments. Is that accurate?
Yeah.
You sure?
Yes.
So, because that's what I think you want. That's what I think you want to come away with.
Yeah.
That's the point you want to emphasize. Our goal really is to see, does the virtual reality environment faithfully mimic kind of natural learning and memory?
Okay. So, I think right now what I'm emphasizing is like the product of what would happen if it were similar, but I haven't compared the two adequately.
Right, but first right, if they are similar,
okay. Yeah.
then that allows you to do, go to your next slide, all this stuff.
Right.
Right? But you're not there yet. So I mean, I think it's fine to say, listen, my goal is really, we need to find out first, are they similar?
Yeah.
And if they are similar, then we can really, once we figure out which cells are turned on or off in response to specific behaviors, we can modulate those cells, their activity via optogenetics and other ways, so that we can see, do these cells really regulate behavior, allowing us to understand the cellular basis of behavior.
Yes.
Right?
Yeah.
So, but I think, go back, I think the point here, and the goal and the kind of the focus initially is VR unless people have already shown it is, but is virtual reality settings, you know, does it faithfully parallel what you see or under normal conditions?
Okay.
Right? And if so, it really might be a powerful tool to figure to assess, to essentially to dissect the cellular basis of behavior. Okay. So go back. And so I think at some level the question is, I mean, so not only are, because you did say it later, I think, how are their brain cells affected, but also does it mimic what we know for normal mice?
Right.
Okay, and then that's what I think you need to bring out this point here.
Okay.
Like, does it mimic? And if so, and so the white in, so that's the hippocampus, that's fine. In B and D, the white, is that the labeling? Is that the activity of the, what's B and D showing?
So this is labeled and this is not.
Where's the label?
So we label, the cells will appear darker in comparison to a brighter background.
Okay. Okay, point them out because it's a little tricky to see. Because what eyes are usually drawn to white, and there's that white in the middle.
Yeah.
So the staining there, is the staining black or is the staining white?
Yeah. So this is black, supposed to be on a brighter background.
So to me that looks like a fluorescent staining. That's not fluorescence?
No.
Okay, so this it's like you use DAB as the substrate?
Yeah.
Okay. And then the cells that are so essentially all cells are activated there?
Yeah. The darker cells.
So it's activated everywhere. I mean, there are a few.
Harder to.
I mean, to me, when I'm looking at that,
Yeah.
I actually initially thought it was a fluorescence slide, and I thought the white was the staining.
Oh.
And the fact that,
Yeah.
because what I see from afar is that there are a lot of little pixelated white dots, but in general, there's a lot of black.
Okay, I see what you mean. There is another set of tissue that I've just stained, which I will try to get better pictures
Yeah,
of.
I mean, that's okay.
Yeah.
But okay, so let's go back for a second. Okay, keep going. Okay, sorry. So this I think is fine, keep going. This just emphasized, I think, the comparative analysis that you want to see. Do we see the activation? Does it, I think, faithfully parallel what you do, what we see normally? And if so, click. We can assess, if so, this gives us greater ability to I think, dissect the cellular basis of behavior. Keep going. Okay. I don't think you need this yet. And so I think, okay, and so go back. So, okay, so what are we doing, right? What's our experimental setup? I think I don't care about FOSS yet. I care about FOSS once I know what your experiment is. Okay, keep going. Keep going. Keep going. Good. This is your experiment, right? Okay. Now, experimental design isn't a great title. The title I would have is, does virtual reality drive neuronal activation? Ideally in a similar way as wild type, but as normal mice. But that's not how the experiment is set up, and that's okay. So essentially, you have mice, right? And they're all basically in that head plate. So the best, if you can convert this into a schematic, it'll be better. It's going to be something like this. I'm not a good drawer. That's a mouse. You're going to split them into three. they're gonna, so have something, this is fun, I mean, if you have something, but this is pretty simple schematic. You could have this, and then you just have them in, you know, have that head plate thing. So have the schematic of mouse in head plate. Okay, and then you can, some of this, like the water scheduling, you don't have to have that schematized there. You say, well, this is water scheduling, and this, all we're going to do is, you know, basically make them really hungry so that the water's a reward, so they will want to do the reward. And then we're going to do VR training. So here's the, now that it gets, to this point, they're all the same, right?
Yeah.
Okay, VR training, one we're going to have control, and that's essentially where they have, they're staring at a blank wall.
They're just not on the VR. They're just in their cage.
Immobilized.
Yeah.
Okay. So they're just in their normal environment, but they're immobilized on a, essentially a ball?
Actually, sorry, no, they're not immobilized. They're just in their cage by themselves. They're placed on the VR room.
Okay, so there, it's control, and it's basically normal environment.
Yeah.
So they're not in a head plate.
No.
Okay. And then VR is head plate.
Yeah.
And then VR1, head plate, VR1, VR2, right? Whatever that is.
Yep.
Right? Image brain for neuronal activity?
Yeah.
Okay. You see how this, we start with the mice, we're going to split them into 3 groups, 1, 2, 3. The first mouse, somehow it's in its cage, you can just have it. It's in its cage, interacting. It's controlled normal environment, no head plate. This one has a head plate, this has a head plate, VR1, 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 same VR1, VR1, VR2. Now our question is, what is the activity, like what is that, how is the brain activity and how does it differ? I'd schematize it something, you know, pictures of a mouse, picture of Mickey Mouse is easy, picture of a mouse in a cage is easy. To get from Google the head plate, you can use yours. VR1, I mean, then you can use, okay. But I think converting that, because imaging and this is kind of a small part of your experiment.
Yeah.
It's really the staining is what's critical.
Yeah.
And then you can say what we're image brain staining is actually neuronal activity we're going to stain FOSS, and you could have images there if you want. But that's how I'd set it up, because this allows you to walk through the logic in a way that they will kind of see a little more the logic of the experiment. Does that make sense?
Yeah.
So something like this, I think, is superior to that. And then you can sit and you can actually have. And you say, well, you know, we're still in the process because some mice I kind of thought it was cute, some mice don't like to learn. It's like, yeah, some people don't like to learn. Actually, all of us don't like to learn in some context anyway. And then what you can do is say something like, well, we don't have any data yet because we're still working on setting up this system. But what I expect, what do you expect to see?
What we would expect to see is that these have the most activated cells. These are in between the control groups are in the 2VR group, and then these have the least.
So least, middle, more.
Yeah.
Schematize it. And then just instead of, you know, this is what we do, and then say expected results. And as long as you say expected results, no one can sit there and say those are your results. I expected results. And you know, you could show, I mean, it's something like that. Do you see how that, and why do we think this? Because this is going to be the most environmentally enriched.
Yeah.
Where this and then this is just they're by themselves, and are they by them? Is it by itself or is it with others?
It's just by themselves.
Okay, they're isolated.
Yeah.
You may want to say control. It's just a normal environment, but or normal cage, isolated, whatever. But something like this, and then it basically brings you right to this, and then you can talk. Well, this is what we'd expect to see, and if we see this, then what?
And if we see this, then we can go on to look at other kinds of experiments that we can do.
Good. Be more precise.
And so now we can go into the sort of optogenetics experiments and calcium imaging experiments that I discussed, where we can inactivate cells and examine behavior and activity, etc.
Inactivate cells and examine how the behavior changes.
Right.
Just be a little more specific.
Okay.
What you say is good. I'm just trying to be precise because the audience is like, it's vague. What do you mean? Well, we can inactivate or activate cells in the same exact VR and assess how the behavior changes. And if the behavior changes, it's really said these cells are helping to drive, to either promote or inhibit that behavior. And that's what I mean by the cellular basis of behavior.
Okay.
Does that help?
Yeah.
Okay. Because I like this part, because that actually helped me. And also when you're talking about the VR, say, because you said they run, I'm like, well, how do they run? You have them freaking in the head plate.
Yeah.
Right, so VR, so what they're in the VR, they're on, just to say, it's not a wheel, but it's essentially a wheel, so they can run.
Yeah.
They can run.
Right.
You know, they're on, it sounded to me like it was this thing that was levitating and they run.
Yeah.
So, you know, when you're, yeah, that's kind of cool. Your classmates probably get although you put some headset. That's all. But I think this is what you need. And send it to me, I can give you comments.
Okay.
But that'll help them understand the experiment.
Yeah.
I don't actually think, go back, I don't think you need the ABC vector stain kit. Keep going. You spend a lot of time on that. I don't think you know, that's how we see if there's staining, but I think for your talk it's really not needed. And because I really think, go back, I actually don't think you need this. I think you schematize it there.
Okay.
Right? Because that's the point that you want to make. And you know, you could say, you know, sometimes depending upon where they're activated in the hippocampus, you can get more or less. It gives us some idea of which cells they may be. I don't know if that's true.
Okay. Yeah, I think I should put this slide earlier because the way we compare the VR and real world environment is because of the FOSS that's released in the real world environment. And so that's how we assess how similar they are.
Right. That point I like. I completely agree that you want to assess how similar they are. That was kind of the point I was trying to make in the first slide.
Right.
Like, is VR a faithful proxy? Is it similar? Are the mice learning in a similar way? Because if they're not, I would argue VR is useless. But they probably are. I mean, I'm not sure if they're, you know, they're learning in the same way if they're stuck at a head plate and all that stuff. But that's an experimental manipulation that probably in the future might go away. But I think go back. I mean, it seems to me this is where you would. It seems to me if you want to make the point, what you should have is this is unactive. This is active. Okay. This is normal, i.e., what we found initially. Okay. Normal. You want VR, and you want to say our question is, well, here's the act, and you get. This is really subtle to me, and I'm used to looking at tissues, not brain tissues, but I'm used to looking at tissues. Right. So you're going to have to, if you're going to say, so here, this is normal, unstimulated, stimulated, or something? What's it?
So the FOSS expressing cells would be labeled darker.
Right. But why are they activated here?
Because the mouse has been
stimulated in some
Sorry, no, we actually stayed for FOS, and this is one where FOS is not stayed.
Oh, okay.
Yeah.
Okay. But when FOS, but why is FOS always active?
No,
It's more active in spatial when it's being
when it's learning.
challenged with learning.
Right.
Okay. So you could say, you know, here, this is where we're challenging it with learning and memory, and this is where we see it.
Mm-hmm.
And our question is in VR, and you know, do we see this? It's a question mark. Right? So I think if you want to say normal VR, do we, bless you, do we see this activation?
Right.
So I mean, I think bringing it up here is fine, but if you're gonna have to point to it and say, you see this dark line, this is where the FOS stain is, and it's black.
Mm-hmm.
So just to explain this, like, we're looking at the sections through the brain. I'm not sure you actually need, I don't think you need this, because this section is this.
Right.
Right, and then this is just like, so I get rid of that, put that here. And you know, frankly, what you could do is you could get rid of this, and you could say, because this is just, when we challenge with learning, this is FOS and it's active.
Right.
And in virtual reality, we think we're going to see this, but you don't know, right?
Right.
Okay. We want to know if virtual reality leads to a similar activation of FOS. If it does, then virtual reality seems like a it's a good system to use to assess learning and memory in kind of a more experimental and manipulable system.
Right.
I think. But I think that would be. But if you bring it up here, I think that would be fine. But I don't think. Maybe I'm wrong. I'm not sure you do that.
Okay. Yeah.
Because the question really isn't. You could just say this black staining, that's FOS, and we see FOS staining when we challenge with the learning and memory task, and we see far less when we don't. Okay, our question is, well, in virtual reality, do we see the same thing? Is the brain doing the same thing?
Right.
If so, we can use virtual reality to really dissect the cellular basis of behavior.
Yep.
So I think that would be good. Then you would just get rid of this, and then have this, and then have virtual reality with the question. Just have a question mark. Do we see that? Because you don't know it.
Yeah.
Okay. And then keep going. But if you do see it, then it makes this. Then you don't need this. Then you don't need this. You don't need this. You don't need that. You go to this. How do we do it? And you have something a little bit more like that.
Right.
Keep going. That's, yeah, and then you just explain the virtual reality. Keep going. Yeah, so here the data, you can, if it's better if you have this on one slide and talk through the data here. Because then you set up the logic of the experiment and it's easier. And if you have the real pictures, it says insert, if you have the real pictures, do it. Keep going. Essentially, you're coming back here to using optogenetics to activate. This is all fine. Everything you said here is fine. Have a schematic over it.
Okay.
And then just talk over it. And the schematic, essentially, go back. Go back to the first slide, your title. Essentially, your question is, once you have it, it's when you're in virtual reality. It's the point you were making. And I don't know how to schematize it. If you have an activity and you can activate or inactivate specific, your goal essentially is to have a specific behavior and activate or inactivate different neurons with optogenetics and see what happens, right? That's the point you want to make on the last slide, but make it with the schematic if you can.
Okay.
And this isn't bad, but you know, what you could do is have, you know, something like this where, and then have have an arrow and say, we're going to activate this side of the brain or the cells here and see what happens.
Okay.
Okay?